Hello, welcome to the first lecture video of Bi 102 Online. This lecture is going to cover Chapter 19, which the subject heading is Systematics Seeking Order Amid Diversity. So this chapter, just an overview, is going to go over how we classify, name, and organize living things into various groups. Humans like to organize stuff. Just think about your dishes cabinet at home or your utensils drawer at home. You probably stack the plates and put the cups in their own place and the forks and the knives and the spoons. So we did the same thing with living things in nature. We like to classify them. So let's get started. So the first thing we're going to talk about is just the science of classifying and naming things, which is called taxonomy. And so you can see the definition here on the screen. Branch of biology concerned with naming and classifying organisms. And on the left-hand side, you can see an example of how some of these might be classified. We can see some examples of animals, fungi, plants, mosses, little protists, and things that you may not even know exist. So historically, the person that we can thank for kind of our modern understanding of taxonomy is an individual named Carolus Linnaeus. You can see he was kind of hanging out in the 1700s. There's a nice painting of him. So sometimes his name is Carl von Linné. He was a Swedish naturalist. We consider him the father of taxonomy. And so he came up with what we call binomial nomenclature, which really is a fancy word that says a two-name naming system. So binomial means two names. Nomenclature means naming system. So what that means is all living things, once they are correctly classified and identified, will have a two-name system. Kind of like we have a first name and a last name. Organisms also have a first name and a last name, and we call this the genus and species. And we're going to see that a little bit later with some examples. Actually, here's one example right now. So this one is called Asclepias tuberosa, which is a scientific name for, the common name might see butterfly weed. There are some rules that go apply when you are using or writing out a scientific name. They have the two parts, so the first name and the last name. The first name is the genus, which is a larger taxonomic group of closely related organisms. And then the second name is the species. And there's a couple different definitions of species we're going to get to a little bit later, but for this definition at the beginning of just the naming system, a species is a group of organisms that can interbreed to make fertile offspring. We call this biological species concept. And the species is the basic unit of taxonomy. So whenever we're talking about organisms, usually we're going to be referencing them as their species. So here's another example. We all know what this is, right? So we have these around our area in the Rogue Valley. So you might know it by a couple different names. So you might call it a puma, a cougar, or a mountain lion. But its scientific name is actually called Puma concolor. And so these scientific names help us be a little bit more specific when we are communicating to people in different regions, where here in Oregon we typically call this a mountain lion. Maybe in the southeast this might be called a puma or a cougar somewhere else. So the scientific names make sure everybody understands exactly who you're talking about. We also are going to have some rules when you are seeing these written out in text, in books or manuals or information or on the internet, or if you are writing them out yourself. So the names are always either italicized or underlined. So they have to stand out from the regular old font in whatever, however they're written. The first letter of the genus name is always capitalized. So that's like how we would capitalize our first name. So the P in Puma is going to be capitalized. But then the second name, the species name, is always lowercase. We do not capitalize this species. So Puma concolor, if you were to see this written in a textbook or on the internet, or as you can see on the presentation today, Puma concolor is italicized, the P is capitalized, and the C is lowercase. And that's the proper way of writing a scientific name. Here's some more examples of some bluebirds and their scientific names and some common names. So we have eastern, western, and mountain bluebird. Now I am not a bird watcher or an ornithologist by any means, so to me they'd all just seem like bluebirds. But to somebody who knows their birds, they would see the slight differences and say, oh, the eastern bluebird would be Cialia cialis, Cialia mexicana, or Cialia Hurocoides. So this is where we kind of get some of those scientific-y Latin and Greek names in there. They are usually descriptive, or sometimes they're named after the people who discovered them, or maybe a location like Mexicana. So now we're going to get into kind of some topics of modern classification, and this is not too modern, but it kind of starts with the idea of evolutionary biology, and Charles Darwin was the originator of kind of this thought. He published his book, The Origin of Species, back in the mid to late 1800s. So his idea kind of sets the stage for understanding of modern classification. So there's a little bit of a background. He kind of proposed that all life is related by common ancestry. So that means we have shared origins from organisms around the world. And so these taxonomic categories reflect that relatedness. So the more closely you are related to somebody, the more closer you are in this taxonomic classification scheme. So like humans to a bacterium, or a protist or a fungus, we have some similarities, but not as close as, say, a human with a chimpanzee, which we'll see some of their genetics here in a little bit. So there's a couple different ways that we talk about modern classification. We have phylogeny, which is evolutionary history. So phylogeny deals with this change over time as organisms adapt to their environment. And then systematics is kind of the study or the science of making these connections, what we call evolutionary trees. So phylogeny is the study of this evolutionary history. Systematics is actually making those connections, actually making a diagram. We'll see some of those here in a little bit to show those relationships. All right, so let's take a look at systematics first. So systematic focuses on building these evolutionary trees, and there's a generic example there on this screen. This is where we get to name stuff. So we come up with the names to name the groups at the different levels of classification. And then we have clades. So clades are kind of these little branches that are coming off. They kind of keep branching and branching and branching. These are all the organisms descended from a common ancestor. So for example, let's see if I can write on this PowerPoint here. So all of these organisms here are derived from this common ancestor. So that would be like a clade. They're arranged in a hierarchy with the smaller clades nesting within larger ones. So this one's a pretty large clade, and so then here's another one. So if I'm gonna, let me pick a different color. So I'll pick green. So if I'm going to do this particular one, this smaller clade is nested within the bigger red one. So within that red clade, they all have some common similarities, but then evolution made some changes. Natural selection kind of diversified a little bit. So while everything in the red circle is related, they split somewhere along the lines, and so then you get some smaller groups that are more closely related. And we have to keep track of that. So systematics allow us to do that. We can look at similarities in anatomical structures. So here's a few examples of the forearm of various animals. We have flying organisms, swimming, running, and grasping. We are humans, so we fit into the grasping category. But if you take a look, they've color-coded the bones that are similar, kind of these homologous structures within, all the way back to pterodactyls, birds and bats, dolphins, seals, dogs, sheep, humans, and a shrew. We all have this kind of arm bone called the humerus. We all have two forearm bones, which is the radius and the ulna. We all have common like wrist-like bones, which we call carpals. Those are the green ones. And then our metacarpals are the pink ones. And then our phalanges are the uncolored ones. So there's anatomical similarities. And so the closer those things are the closer those anatomical structures are to each other, most likely the closer they are related to on, say, an evolutionary tree. And then also here we have some microscopic anatomical similarities. You have to get under the microscope to see these. Radula are little kind of teeth, things that you might find on a snail or a slug. Bristles you might find on an insect or a worm. And pollen grains are structures, obviously reproductive structures of plants. So historically, we were able to use what we saw, right? So we can dissect animals and we can look at their bones and compare them. But then as technology increased, we were able to look at things under the microscope. So what we see first, we had all of these classifications, and as technology and knowledge increased over the decades and centuries, we were able to make better and better classification groupings because we could get more information about our organisms. All right, so here's just a couple more examples of some microscopic similarities of pollen grains. Then, like I just mentioned, with our increasing technology and knowledge, we can start comparing things at the molecular level. And this is really where taxonomy is at this point in our scientific communities. Yeah, we can look at anatomical features or what what's called the phenotypes, the expression of traits are, but really we have the science and the ability to easily now look at the DNA and the RNA and the molecular comparisons to see how things are staying the same through evolutionary time or are making these splits and diverging through evolutionary time. So they help to reconstruct your phylogenetic trees. We're looking at similarities between DNA nucleotide sequences and even chromosomes. So if you take a look at the picture of the right, this is a picture, a representation of human chromosomes. This is our DNA, all the instructions that make us us. So the H's stand for the human chromosome, and the C stands for the chimpanzee chromosome. And if we're to look at all of the DNA between the humans and chimpanzees, we share about 96% of our genes, our instructions to make us who we are pretty much the same as a chimpanzee. So we're different by about 4%. So we can see that in the comparison of the chromosome structure, and also the little banding patterns that you can see on the chromosomes represent similarities in genes that we have, both us and chimpanzees. So lots of similarities, difference enough to make us unique. All right. So then, as we're on this discussion of taxonomy, there are different groups that identify the relatedness of different organisms. So think of these as like stacking bowls or like decreasing or increasing sizes of boxes. So the biggest box is going to be the one at the very top of this colorful rainbow picture here on the left-hand side. So the biggest group is called a domain. So the domain is going to include all of the things that have like the most basic things in common. And then within that domain, there's going to be smaller groups that we can separate. Okay, so everybody is, a eukaryotic organism. So hopefully if you took Bio 101, you remember what a eukaryotic cell is. It's a cell that contains a nucleus. So of all of the organisms that are eukaryotic, we can break those into smaller groups, right? So I have a nucleus in my cells, so I am a eukaryotic organism. I am in the domain Eukarya, but so is a oak tree, and so is a blue whale. So there has to be some way we can further break that down. And that's what all these taxonomic hierarchy ranks do. So they have these ever decreasing kind of more and more specific characteristic categories that we're going to put organisms in. So this is where we get down to eventually that genus and species that Linnaean taxonomic binomial nomenclature allows us to do. So if I get my marker back out right here, so genus and species are the last two ranks of this taxonomic hierarchy that's going to give you the individual organism, right? So the Puma concolor for the mountain lion. It's nested. Each level includes all of the other levels below it. I clicked too fast on that one, sorry. So we're going to talk a little bit about these domains. So there's, of all of the living things that we know on this planet so far, we have identified organisms and put them into three big domains. We have one that is called bacteria, we have one that is called archaea, and then we have one that is called eukarya. So you can see there's a common ancestor. Again, we do not know the history. We don't have record of who that common ancestor is or was, but we can see we kind of all branched off very early on in the history of living things. So bacteria is one group. They have enough unique characteristics that they get into their own branch of this tree of life. Archaea have enough of their own differences that they get their own branch on the tree of life. And then eukarya, they have enough of their own differences to get their group. So our three domains are bacteria, archaea, and eukarya. Now again, linking back to your general bio or whatever, if you took cell biology, Prokaryotic cells are single-celled organisms. Their DNA is not bound in a nucleus, and they do not have membrane-bound organelles. So those are some key characteristics. So bacteria and archaea are both prokaryotic in cellular structure. So you might be asking yourself, well, why are bacteria and archaea, why aren't they in their own domain if they're so closely related in being prokaryotic cells? Because they actually used to be. All single-celled organisms that were prokaryotic were in their own grouping. They were just called prokaryotes. But then again, as science and technology and knowledge has increased over the years, they were able to find enough differences between archaea and bacteria that they're like, these guys, even though they're single-celled and prokaryotic in nature, there's enough differences that they get their own domain. And then eukarya on that third branch, these are all the organisms that their cells are eukaryotic in nature. So remember some of those characteristics. They have a membrane-bound nucleus, membrane-bound organelles, they're larger in size, they're complex, all the fun stuff from way back in your general bio class, your cell biology class. All right, so those are our 3 domains. So here's some representative pictures, very generic pictures of a bacterium and an archaean. So these are 2 domains. They're kind of boring looking, even though I teach microbiology as well, so I'd argue that they are not boring in themselves. They are really fascinating organisms, but really they're just single-celled, no membrane-bound nucleus, and they just live to replicate and divide and live and replicate and divide. So kind of a boring lifestyle, but they do some pretty cool things. Then of the domain eukarya, this is the domain that we are probably most familiar with because a lot of these organisms we can see with our eyeballs, and we ourselves are in the domain eukarya. So this includes things like in the picture, insects, fungi, protists, mammals, flowers, plants. Okay, so do you remember that big kind of that nesting picture that we had a couple slides ago, that kind of the rainbow order? So it went kingdom, phylum, class, order, family, genus, species. So the domain eukarya, it's going to split into 4 kingdoms, right? So that of all of the organisms that have a eukaryotic cell within their cellular structure, we have divided all of those things into 4 smaller groups, which was the next one down, and that's going to be kingdoms. So we can split everything that has eukaryotic cells as their cellular structure into animals. We are of the kingdom Animalia. Plants or plantae, that's things that you are familiar with as being plants. We have fungi, so that's like mushrooms and yeasts and things like that. You're probably familiar with that. And then lastly, we have what I call like the junk drawer of eukaryotic things. It's called protista or protists. It's everything that's not clearly an animal, a plant, or a fungus. So usually these are going to be things like amoebas and paramecium, single-cell protists, but they also include things like multicellular algae, seaweeds, those types of things. Okay, so now that we're going to focus primarily on the eukaryotic domain. So the kingdoms, animals, plants, fungi, and protists. So we mentioned archaea and bacteria. They do exist, but that's not going to be the focus of this particular class. We're going to focus more on the eukaryotic organism. All right. So the last couple questions we're going to ask is, why do they change? Like, why are there differences? And you may not know that there's been changing, because I don't know how much taxonomy you came into this class with. But just as I've mentioned a couple times already, information, we learn more stuff. We can now analyze DNA. We can see how closely related things are over time. And sometimes we might see groups split, and we have to come up with new classifications as we learn more and more and get more information about organisms. So we may have at one point just said a giraffe is a giraffe because it looks like nothing else. But then as they did more genetic analysis, they actually broke the giraffe, the African giraffe, into 4 different groups and some subspecies within that. All right, so we get new information. Top levels rarely change. So something in a eukaryotic domain is not going to switch to a prokaryotic domain, or an animal kingdom is not going to switch to a plant kingdom. So those top levels, those are pretty kind of set in stone. But as you kind of get down into the phylum, class, order, family, genus, species, those things might shift as more information comes along, because we're now in those really small, narrow classification categories. And if we find something that doesn't quite fit, we've got to find a new place for it. Species changes a lot. That's probably the one that changes the most. African elephant has divided into two different species, the savanna and the forest, and once they were all just called African elephants. And giraffes have been divided into 4 different species in these different groups. So just a few examples of some changes that we've seen in recent decades. All right, so since we're talking about species and that's the one that changes the most, what is a species? We know it's the last category in our, classification, but what does it actually mean? Well, that's an interesting question, because you ask, 15 biologists, you might get 15 different answers as to what a species is. So we're just going to break it down in kind of 2 large definitions, kind of a historically accepted definition of a species and a more modern definition of what's accepted as a species. So this first one is called a biological species, a concept. This is how I learned it when I went to undergraduate school to get my biology degree. So it is defined as a species is a group of individuals that are capable of interbreeding and producing viable, fertile offspring. So what that means is 2 organisms can mate, and their babies can grow up and mate and make more of that same organism. So if they can do that, those organisms that mated, those first 2 parents, they would be the same species, and their offspring would be of that same species. Okay, so if horses, so in our example here on the screen, horses mate with horses, they could make more horses. Donkeys mate with donkeys, they can make more donkeys. So those are different species. But you may be aware that horses and donkeys can mate, and they end up with an offspring called a mule, right? So it's got kind of in-between characteristics between a horse and a donkey. And that's fine. The mule is viable. It will be alive. It won't be, miscarried due to genetic abnormalities. But mules cannot mate with other mules to make baby mules. So they are infertile. So the mule does not count as a species because it cannot mate and make more fertile offspring. So the only way you're ever going to make a mule is you crossbreed a horse and a donkey. So according to the biological species concept, horse is a species, donkey is a species, mule is not a species. Okay. Sometimes it's easy to apply to a horse, and donkey, and mule example, but what about things that are living in the bottom of the ocean? Or we might, some things that are microscopic and it's hard to tell their characteristics and how they mate, and are their offspring viable and fertile. And we have to remember there's a whole slew of organisms out in the world that do not reproduce sexually. So how would this apply to an organism that reproduces asexually, just making a clone of itself? So it was good at one time. It does not apply as we're learning more and more information and identifying more new species in our world. Like bacteria, archaea, and protists reproduce asexually. So this we cannot even apply to those organisms. And there's a lot of those out there. So a more common, modern, I should say, species concept is called the phylogenetic species. And I know this chart on the left-hand side looks crazy complicated, and you don't need to learn that. It's just there as an example to show you how we get this phylogenetic concept of a species. Because we talked about these phylogenetic trees, the clades, they kind of analyze the genetic information, that molecular relatedness, and they're finding your individual species that way. So it defines a species as the smallest diagnosable group that contains all the descendants of a single common ancestor. So I'll get my little pen up here. So I'm just going to go from the very top. I think this is plants. Angiosperms are flowering plants. So right here, angiosperms is this bracket right here. So all of those things that came off of angiosperms is going to be related because they are all seed-producing flowering plants. But we know there's a whole bunch of those, and they've branched and they've separated and they've branched. So if we go all the way up here to the very top, Arabidopsis thaliana would be one species. And so everything, I should pick a different color because that's green. So everything in this line here, that's going to be a single species. Now, if we look at its neighbor, Arabidopsis liriata, it's got its own line. That's a different species. We'll notice they're related because they have the same genus name because they came off of this branch right here. And so they are related, but they're not exactly the same. We don't know if they're going to crossbreed. That's not part of this definition. But each one of these organisms that have their own line, that represents a single species. Okay. Now this is a better application of the definition of species because it can be applied to sexually and asexually reproducing organisms. We don't have to leave out a huge slew of organisms because we do not, they don't match our definition of what a species is. Okay, and it's starting to, replace that biological concept of a species in textbooks and just in the general scientific community. All right, so as we're wrapping up our discussion, we might ask, well, how many species actually exist? Have we counted them all? Have we found them all? And the answer is no, we have not. So as far as the amount of organisms on this planet, if we're to collect all of them together in our ecosystem, our biosphere of the planet, or the individual ecosystem like a rainforest or a tundra or a temperate forest or a desert, we call this biodiversity. So the total number of species in an ecosystem. The higher the biodiversity, the more organisms you're going to find in that area. So we have about a little less than 2 million named species. So these are the things that are in the books, in the taxonomy books. They have been recorded, identified, named. They have a place in that taxonomic hierarchy. However, this is a little bit biased, right? So in the parentheses, it's biased because most of the organisms that are classified are big because we can see them, right? And most have been identified in temperate regions. And if we think about, I have a big map on my wall here. If we think about where people live, a lot of people, especially, historically speaking, in the scientific enlightenment period of our history, lived in temperate regions. So we as the human species ourselves, we as humans in the past, as we were learning to observe and start recording this, as the study of science started to develop, we looked around us and we recorded those things. So a lot of what we have on record is what we just interact with. So there's big gaps in our knowledge based on a couple different reasons. So of the 1.6 million organisms that we have identified, only about 5% are these microscopic prokaryotes and protists, about 20% plants and fungi, about 75% are animals, which is probably not even close to the actual number. There are probably way more prokaryotes and protists out there in the world that are not represented in this 1.6 million number. It is estimated there might be around 8.7, somewhere between 7 and 10 million currently existing, yet to be identified. We find about 15,000 new species identified every year, and the tropical rainforests are thought to be home about 60-something%, about 2/3 of existing species, most of which are unnamed. Tropical rainforests and coral reefs are some of the most diverse ecosystems on this planet, and we have a hard time getting in there and identifying them because, going underwater is hard to do. Going in a dense rainforest is hard to do. So there's going to be a lot of things still waiting for us to discover them. So continue on answering our question. There's only been about 5,000 species of prokaryotes, archaea, and bacteria that have been officially named, but you take a soil sample, you might find 4,000 different species in just one soil sample. So clearly there is undiscovered organisms, just we don't have the human manpower to find all of these things. It takes time to take samples and grow the organisms and test them genetically and microscopically. For these bacterium, these prokaryotic organisms, they would be considered a different species as long as 30% of their DNA is different from another bacterium. So there's different criteria on how we speciate these microscopic organisms like bacteria. Another thing we need to think about when we're trying to put a number on how many organisms we have named and classified is how easy is it to find them, right? If they're super small, it's going to be hard. Maybe they're super similar. We haven't found enough differences to maybe put it in a new species. So maybe there's even a subspecies or a strain. It's related, but it's not quite exactly the same as all the other samples you have. How easy is it to find the organism? You see a unmanned rover here going to the bottom of the ocean in this picture. That's really expensive to do that, and we don't have a ton of people wanting to do that. To be part of this exploration. And so we're finding a lot of new species because there's like a whole bunch of the ocean floor that's not even discovered yet and mapped out. So that's a big one of those empty boxes of where there could be organisms living out there that we've just never even seen before. How many people are educated enough to go out and actually find and classify these things? And so here, just to leave this last note here on this slide, the deep sea floor might have hundreds of thousands of unknown species to be discovered. We've only mapped and identified about 5% of the ocean floor, and the ocean covers 70% of our whole planet. So like I said, lots of unknowns in the ocean. And then lastly, just a story. This is the black-faced lion tamarin. It lives in the rainforest in the eastern coast of Brazil, looking at my map over here in the rainforest. One of the drawbacks of classification is sometimes organisms might have gone extinct before we even identified them. This one didn't, but it wasn't recognized scientifically until 1990, and it was already put on the endangered and conservation lists in the early 2000s. So within a decade of its discovery, we're already on the brink of maybe even losing this species. So there could have been species that we just never even knew existed and got bulldozed under in some, changing a rainforest into a farm. So we don't know about that. All right, so that wraps up our first chapter 19 lecture video. So please make sure you're reading the chapter, going through and using the note outlines and the study guides to help you review the material. There's supplemental videos on the Blackboard shell that you can watch to help you reinforce these things, and make sure you're working on your homework to get that done. All right, so I will see you next time. Bye.